The thalamus has widespread connectivity with the cortex, facilitating transmission of sensory inputs to cortical regions and intracortical communication. Understanding thalamocortical organization is therefore key to understanding whole brain dynamics. Previous work has largely tried to achieve this by focusing on the organization of thalamic nuclei, yet the principles underlying the cortical patterning of thalamic projections remains unclear. A recent study in mice found thalamic nuclei form a hierarchy arranged along a medial-lateral axis of gene expression. Notably, position along this axis was associated with differences in cortical projections, suggesting it represents an underlying principle of thalamic organization. We therefore wanted to see if the results of this mouse model would extend to the human model as well. To do so, we first defined seeds throughout the thalamic volume. We mapped gene expression data from the Allen Human Brain Atlas to each seed, building a matrix of seed by gene expression values. From each seed, we also conducted probabilistic tractography to 250 cortical regions, building a matrix of seed by cortical connectivity values. We combined this with the gene data and performed a principal component analysis, yielding us a matrix of principal component scores, which tell us how strongly each seed relates to the underlying component, and principal component loadings, which tell us how strongly each gene and cortical region defined each respective component. We can use these to answer if the dominant axis of variation maps onto a medial-lateral thalamic gradient and what patterns of cortical projections emerge from it. Returning to our principal component score matrix, we want to extract scores for the first component, which in our case explains 30% of the variance. Projecting these scores onto their respective seeds and surrounding voxels, we see a medial-lateral gradient emerge, which we can further confirm by correlating each seed's medial-lateral position with its principal component score. Next, we check to see if similar genes in the mouse and human were associated with this gradient. Using the first principal component loadings for the genes, we find that the genes with the largest positive correlation with this axis are enriched for genes most highly correlated with the mouse axis. Conversely, genes with the largest negative correlation were enriched for mouse genes, although not to a significant degree. Finally, we can project the first principal component loadings for cortical regions onto the brain to see which areas are associated with connections along the medial-lateral thalamic gradient, finding that these connections map to an anterior-posterior cortical one. Furthermore, we wanted to know if this cortical pattern corresponds to cortical function, especially as thalamic connectivity has previously been associated with cortical intrinsic timescales. Indeed, we found that intrinsic timescales were highly correlated with our cortical gradient. Interestingly, these thalamic and cortical gradients also correspond to known patterns of neurogenesis, raising the possibility the properties of these gradients are shaped by early development. In summary, the principal axis of transcriptomic and structural connectivity in the human thalamus occurs along a medial-lateral gradient, which is potentially conserved across species, is associated with distinct cortical patterning and function, and may also reflect sequential development.